Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Here we solve questions from IIT JAM examination. As you know, this is the entrance examination to get admission in the MSc in IITs. So we'll have a question from the IIT JAM paper of biotechnology. The technique that involves impacting samples with electrons is. The options are NMR spectroscopy, ESI mass spectroscopy, IR spectroscopy, and another is UV visible spectroscopy. This is a 1-mark MCQ type of question from paper of biotechnology 2019. So let's solve this question. Yes, as always, let's have a look at each and every option. So first, starting with NMR spectroscopy. This NMR spectroscopy stands for nuclear magnetic resonance. See here, when we say spectroscopy, always there is a use of one of the wavelength from the electromagnetic spectrum. If you remember electromagnetic spectrum, it is having the lights of different wavelength. Like there is a UV light, there is a visible light, there is a infrared, there is a microwave, there is a presence of X-rays, right? So all these having different wavelength, and according to their wavelength, their intensities are also varying. So the thing is, according to our purpose, we use different wavelength to solve our problem. Like NMR spectroscopy, we use it to resolve the protein structures. The DNA structure, that is also can be resolved using NMR spectroscopy. X-ray crystallography, that is also used to find out the DNA structure. So according to our requirement, we use the different electromagnetic radiation. Likewise, now try to understand the NMR spectroscopy. So as we say, nuclear magnetic resonance spectroscopy. In this spectroscopy, there is a use of radio waves. Here we use the radio waves, and these radio waves are impacted on the nucleus in presence of magnetic field. So what happens after this? This nucleus is having its own spin. Initially, say it is having alpha spin. After this treatment, this nucleus changes its spin to beta spin. Okay? And when the nucleus is coming back from beta spin to alpha spin, it emits the energy. That is what we record in the NMR spectrum. So here there is a use of radio waves as well as a magnetic field. Now, the another thing in NMR spectroscopy to be remembered is how much that nucleus is covered with the hydrogen atoms. Okay, so as you know, the hydrogens, if we say the structure of methane, that is a CH4. So there are 4 hydrogens are there around the carbon. So this is like a shield. The carbon is shielded with the hydrogen. So as much as number of hydrogen atoms are there around the carbon, there would be a less peak. That is acting as a shield. That hydrogen atom doesn't allow this magnetic field radio waves to act upon the carbon nucleus. And hence we will get less spin as the number of hydrogen atoms are more. In this way, these peaks are recorded, and using this peak we can find out the structure of that particular molecule. Okay, so this is about NMR spectroscopy. Now, in reference to our question, the question is asking impacting electrons, and here we are not impacting any electron in the NMR spectroscopy. So this is not the correct answer for our given question. Now go to the next option. Yes, second option is ESI, that is electrospray ionization. This is related to mass spectroscopy. Mass spectroscopy uses mass to charge ratio to find the molecular weight, to determine the molecular weight of the compound. Okay, so what happens here, in the mass spectroscopy there is need of formation of ions. The ion formation is needed. For the formation of ions, there is a bombardment of electrons on the macromolecule, and thus macromolecule get converted into cation and it forms in a gas phase. Gas droplets are formed having the ions in that. Embedded ions are there in that droplets, and these ions are further used to determine the molecular weight. So here there is a reference of electron present, and actually this is the answer for our question. Now we'll have discussion for other two options also. Let's understand what are other two techniques given in the option. The third type or the third option given in our question is infrared spectroscopy. This is a type of vibrational spectroscopy. The energy which is present in infrared radiation is sufficient to have the bond vibrations. It can give stretching vibrations or it can have a bending vibrations in a molecule. Due to the bombardment or due to the application of infrareds on the molecule, there may be a bending vibration or there may be a stretching vibration can be observed. Secondly, these vibration varies from bonds to bonds. See, in chemistry we study that there is a single bond, double bond, triple bond, as well as in biomolecules also there is a presence of different single or double, triple bonds are there, right? So these molecules will have different absorption of infrared rays according to their bonds, number of bonds, single, double or triple. So this spectroscopy is used to find the functional group in a molecule. Also it is used to find the bonds. How much bond means single, double or triple bond is there in a molecule. So this is the application. Now in reference to our question, there is a involvement of infrared radiation here and there is no reference of electrons. So this is not a correct answer. The last option is UV visible spectroscopy. The name itself is indicating that there is a use of UV visible radiations. So what this radiation does on a molecule, these are exactly equal. These are having exactly equal energy which is required for electron to jump from lower level to the higher level. Okay. So when the molecule is bombarded with the UV visible light, there is a transition of electron. Here the electron transition is observed and hence there is a transition of electron from one atomic orbital to another atomic orbital. This is there in a UV visible spectroscopy and the source uses UV visible light. So again there is no reference of electrons, right? So this is also not the answer for this question. So the answer for this question becomes option B, that is ESI mass spectroscopy. This is the correct answer. Here is a next question for next video. This is a 2-mark MCQ type of question from paper of Biotechnology 2019. See what is the question. Solutions of the following peptides are prepared separately at a concentration of 1 millimolar. Among these 4, which one has the highest absorbance at 280 nanometer? See, different combinations are given of the amino acid, different peptides, 4 peptides are there and having a different combination of amino acid. And the question is asking which will have the maximum absorption at 280 nanometer. So we'll solve this question in my next video. Till then, don't forget to like, share and subscribe my channel. Also to hit the bell icon. Thanks for watching. Bye.